Welcome back. Science of the Cell Blueprint. I'm not going to lie, I'm a little bit excited about this one, and the reason why is in the levels of organization video, I talked about how cells make up an organism, that cells lead to tissue, tissue leads to organs, organs lead to organ systems lead to the organism. Therefore, the healthier the cells are, the healthier the organism is. I think, made the point, or the argument at least, that a healthy organism makes a better member of the population or society or a community. Also, if an organism, a human being, is not feeling well, it's some kind of cellular dysfunction. Cells make tissues, tissue make organs makes organ systems, and at some point in there, symptoms or signs are going to develop. So when somebody has symptoms and they're not feeling well, they have cellular dysfunction, period. Then in the next video, or the last video, we talked about if cell dysfunction is what leads to organism dysfunction, human symptoms, disease, not feeling well, then what causes cell damage? Because if you damage cells, you damage the tissue, damage the organ system, and then the person. What causes cell damage? And we talked that there's major two things, if you remember. There was stress maladaptation, that we're constantly exposed to stress, things like temperature and exercise, and sometimes it's positive and sometimes it's negative, or if it's too much or too extreme or too chronic, that you get maladaptation due to the stress, and you can get things like cell size changes, hypertrophy or atrophy or hyperplasia, metaplasia, dysplasia, that there are physiologic changes that can occur with a cell, and that's positive and normal. But there's pathologic ones as well. We also talked about it's not just cell or stress maladaptation, but directly acquired cell injury or cell dysfunction or cell damage. Things like physical trauma or chemical insults or micronutrient deficiencies, or in some cases excess. Perhaps it was an infection of some kind. Now, that was what causes cell dysfunction. Now in this video, we're going to talk how cells become dysfunctional on a biochemical level on the inside. And this is where I get really excited about this. All right. So I guess, well, let's just go ahead and get started. So some of this presupposes a little bit of knowledge of biochemistry and physiology, but I'll walk through some of these things so that you kind of have an understanding. Let me say this. So at the end of this video, I'm going to show you, actually, you know what, I'll just show it to you right now, why not? There's a list of things that is said to cause cell injury or cell dysfunction. Mechanistically on the inside. And here's just a short list of them. Things like ATP depletion, mitochondrial damage, calcium entering into the cell, too much, or I would even argue too little reactive oxygen species or free radicals, damage to the membrane, or possibly misfolded proteins. Now, here's the thing. That's a list that you might see. But what I'm going to show you now is it doesn't matter where you start one of those things. There's a domino effect, and if one of those things happens, all those things happen. So once again, the thesis is that if you have cell dysfunction, you're eventually going to lead to some kind of human dysfunction or disease or signs or symptoms, and that if somebody, one of your patients or clients, is not feeling well, that it's because they have cell dysfunction, and this is what is going on inside of the cell that makes a cell dysfunctional. All right, so let's just start, and it doesn't matter where you start. I'm just going to start with suboptimal amounts of ATP. Now, you probably know ATP is adenosine triphosphate. It's said to be the currency or the energy in the cell, but to do what? Is for the cell to do what that cell does. If it's a neuron, ATP is responsible for allowing that neuron to fire. If it's a muscle, ATP is responsible for allowing that muscle to contract and relax. If it's a parietal cell inside the stomach, ATP is what's responsible for allowing that parietal cell to make hydrochloric acid. So ATP is the thing that is used for a cell to do what that particular cell does. So what happens if there's not enough ATP? So I'll just start. It's suggested that about 5 to 10% depletion of ATP is threatening to this particular cell. And that doesn't sound like very much. So up here we have this thing called the sodium-potassium pump, or the sodium-potassium ATPase. This is found on the cell membrane of every single cell in the body. It is said to be responsible for about 20% of your basal metabolic rate, which is kind of a lot, because of its consumption of ATP. Now that ASE is an enzyme of some kind, and what this means is that it's an ATPase. It's going to break down ATP in order to transport things like sodium and potassium, in this case, across the cell membrane. They can't cross by themselves. They need some help. Anytime you see ATP involved, it means it's pushing it against a gradient. So here inside of a cell, and if you don't know cellular biology, I encourage you to look at some of the other courses that I've done, level 1, level 2, biochemistry, physiology. But the sodium-potassium pump, or ATPase, is going to pump against a gradient sodium outside of a cell, 3 of them, sodiums, and 2 potassiums inside of a cell. So when this thing's moving along and there's plenty of ATP, it takes 3 sodiums and puts it outside of the cell, and takes 2 potassium to exchange it and put it inside of a cell. Now that causes the resting membrane potential or transmembrane potential. We're not going to talk about that here. But the key is that it needs ATP to do so. And magnesium, incidentally. Anytime ATP is there, so too is magnesium. So what happens if somebody lacks a little bit of ATP? Well, if this relies on ATP in order to pump sodium against a gradient outside of a cell and bring in potassium inside of a cell, then you get less of that. And then what you do is, problem number 1, if you lack ATP, is you start to get sodium accumulation inside of a cell. Now what that causes, too much sodium inside of the cell causes an osmotic gradient that tends to draw water inside of the cell. And in level 3, physiology, I present a paper that says small alterations, small fluctuations in cellular hydration, either too much overhydration or too little dehydration, which is to say cell swelling, overhydration, or cell shrinking, dehydration, leads to pretty significant and major biochemical alterations, as well as alterations in gene expression. I'll say that one more time. This paper said small alterations or fluctuations in cell hydration, swelling or shrinking, causes fairly major alterations in biochemical pathways as well as with gene expression. And that sodium accumulation inside of a cell that leads to water coming into the cell, cell swelling, hyperhydration, is said to be one of the first of this domino effect alterations causing cellular dysfunction, which is all what we're talking. How do these cells become dysfunctional? This is, if you're a practitioner, this is what we're trying to do. Fix the cells. Because if you have healthy cells, you have healthy tissues. If you have healthy tissues, you have healthy organs. If you have healthy organs, you have healthy organ systems. And if you have healthy organ systems, you have a healthy organism who probably is a happier, healthier, more productive person in the population, hopefully. All right, so number 1, you lack ATP a little bit, so you get a sodium accumulation inside of a cell that tends to draw water towards it. You get hyperhydration, cell swelling, and that's the first step, most common step in cellular dysfunction. But that's not it. If the cell starts to swell, for example, okay, and we no longer are making ATP. I'll say this, we're no longer making enough ATP for some reason, but we're still using it. These are still using it. Over here, I'll show you what this does later, still using it. Remember that really old pathway called glycolysis? Glycolysis is the biochemical pathway that takes glucose, splits it into 2 pyruvate molecules in aerobic respiration when there's oxygen present. And if you remember, glycolysis happens in the cytosol of every cell in the body. It can be regulated. It can be sped up or slowed down. And if there's a lot of ATP, then that's a period of energy abundance. And in these periods of energy abundance, then we will inhibit glycolysis. The major regulating enzyme of glycolysis is called phosphofructokinase 1. And lots of ATP inhibits this enzyme. Because if the mitochondria is making a lot of ATP, we don't need more. So we can take glucose and put it somewhere else, like store it as glycogen in the appropriate cells. But there's something in biochemistry called the ATP-AMP ratio. ATP is adenosine triphosphate. There's 3 phosphates on it. And if it's used up, it loses a phosphate to become ADP. And it can lose another phosphate to become AMP. So follow this. A lot of ATP and a little bit of AMP. That means we have lots of energy, an energy-sufficient state. Let's store things. Let's block glycolysis. Let's take that extra glucose and make it into glycogen because we have so much ATP right now. ATP means a lot of energy. AMP means we don't have any energy. We need more. A lot of ATP, a high ATP to AMP ratio means storage. We have enough. We don't need more down here. We need more here. Let's inhibit glycolysis. On the other hand, let's say we have some amount of ATP depletion. Now we have ATP depletion. We're not making as much ATP, but we're using it. So we're using up the gas, ATP, and then our pile of recycled ATP, which is now AMP because it was used. So the ATP-AMP ratio gets more narrow. Now, when we have more AMP, what happens is ATP inhibited glycolysis via phosphofructokinase 1, but high amounts of AMP stimulate it. So let's go back. We're talking about a dysfunctional cell. A dysfunctional cell that for some reason, we're going to wrap this up towards the end, you'll see, for some reason is not making enough ATP. There's a whole bunch of reasons for this. Not making enough ATP. One of the things that happens is you get cell swelling. Why? Because you don't have ATP to run the sodium potassium ATPase, so then sodium doesn't leave, it stays inside of the cell, and because all that sodium is inside of a cell, causes a gradient, an osmotic gradient that tends to draw water inside of the cell. Now we have water inside the cell swelling, biochemical pathways might be inhibited or stimulated, and gene expression might be altered as well. Another thing that happens is if we're no longer making ATP, but we're using it, because a lot of these things will continue to use it, then our AMP levels will go up. ATP goes down, AMP goes up. Now we have more AMP that's going to stimulate glycolysis. And as you will see, if we're lacking oxygen, which is a part of this process in a second, if we don't have oxygen, if we're not making ATP, it's probably because the mitochondria is not working real well. And if the mitochondria is not working well, then pyruvate has nowhere to go. Aerobic glycolysis means that pyruvate ends in pyruvate and goes into the mitochondria for the citric acid cycle, electron transport chain, making ATP. But let's say for some reason the mitochondria is not working well, or there's not enough oxygen, or the citric acid cycle is being inhibited for some reason. Well, pyruvate doesn't have anywhere to go, and so instead it goes in anaerobic glycolysis towards lactate, which is fine. Lactate is cool. It was discovered, I think, in 1780 or something like that. 200, and we're still learning more about it. Some people say it's a hormone. Anyhow, so now we have lactate, but what is known about lactate, or thought to be about lactate, is when you make more lactate, you change the pH of this cell. You make more hydrogen ions, which means you increase the acidity, which means you lower the pH. And the role of pH inside the cell is for enzyme production or enzyme function. And if you lower the pH or increase the acidity or increase the hydrogen ions, it doesn't matter how you say it, you change the pH of the cell, enzymes are not going to function as well. What are we talking about? Cell dysfunction. So if you lack ATP, if it's not being made but it's being used up, you get higher amounts of AMP, you start to inhibit glycolysis, stimulate glycolysis, which incidentally, if no glucose is coming in, then you're going to start to use more glycogen. You stimulate glycolysis. And because pyruvate has nowhere to go, because it's not making ATP, because the mitochondria is not working well, then you're going to end up with an accumulation of lactate, which changes the pH, lowers the pH, increases acidity, and now we start to get enzyme dysfunction. You can see how this is a dysfunctional cell, and as we talked about in the previous video, cell dysfunction, if it's whatever is causing it's not removed, leads to necrosis or cell death, which leads to tissue dysfunction and necrosis and so on. Okay, so that's from glycolysis. So if we're not, now we're in anaerobic glycolysis, which if you remember from level 1 biochemistry, only makes net gain of 2 ATP, which is not enough to run the cell. So now we're really starting to lack in ATP. There's another ATPase, ATP-dependent enzyme, cell membrane, called the calcium ATPase. Now this is where things start to get really bad, is this also requires ATP. Now, calcium, intracellular calcium, needs to be very tightly regulated because, as you see in a second, intracellular calcium stimulates a whole bunch of pathways, a whole bunch of enzymes that, if they're not regulated, cause major cellular dysfunction. So where are we? We're lacking, all we talked about so far was a lack of ATP. Remember that list I showed you at the beginning? The only thing we're talking about right now is ATP depletion. That's it. If we lack ATP, then we lack the ability. This calcium ATPase is a calcium regulator for the cell. That if there's too much calcium, then this ATPase is going to take that calcium from inside of the cell where it can cause damage and put it outside of the cell. Now, it's suggested that the ratio of extracellular to intracellular calcium can be as much as 100,000 times more calcium outside of a cell than inside of a cell. This is an active transporter because it's using ATP. That's a mound of calcium outside of a cell and only a little bit inside of the cell. And now normally the body across the cell membrane likes things to be normal or equal, homeostatic. But this is what is typically the case. A little bit of calcium inside of a cell, loads of it outside of the cell. And this is this shoveling more calcium from inside of the cell onto this huge mound outside, against the gradient, against what the body desires, and it requires ATP. But guess what? If you lack ATP, then you can't run the calcium ATPase. And so now we can't pump excess calcium from inside of the cell to outside of the cell, and we start to get an increase in intracellular calcium. Where was that extra calcium coming from? It turns out that there is what's called a sodium-calcium exchanger, or sodium-calcium, what's also called a countertransport or antiport. Now what that simply means is, under normal physiologic conditions, this is another calcium regulator. You can see how much the cell does not want extra calcium. Now what it normally does is, remember, let's take a step back. The sodium-potassium pump normally pumps sodium outside of the cell. So there's a whole bunch of sodium outside of a cell and less inside of a cell. That's normal. In normal physiology, and the majority of sodium is outside of the cell, that this sodium-calcium exchanger can scoop up some of the extra intracellular calcium, if it has too much, scoop up some sodium outside of the cell where the majority of it is, and then flip it. And so it takes the calcium inside of the cell and puts it outside, and brings sodium from outside of the cell inside, which isn't a problem, because when it does that, if it brings sodium outside to inside, it gets pumped back out, the properly functioning sodium-potassium pump. I'll say it again. In normal physiological conditions, you have excess sodium outside of a cell that really wants to come in because there's a gradient. If you get a little bit too much intracellular calcium, this exchanger can scoop some of that intracellular calcium and extracellular sodium and swap it. And now calcium's outside of the cell, safe, not going to cause any problems. Sodium just came into the cell. That's totally cool because the sodium-potassium pump takes 3 sodium ions and pumps them out for every ATP that it has. That's great. So you can see how that normally works. But research has suggested, or shown, that this sodium-calcium exchanger can reverse itself. So what that means is now, if you don't have a lot of ATP or have ATP depletion, then the sodium-potassium pump's not going to work. You're going to end up with intracellular sodium. Now, because you have intracellular sodium, that sodium-calcium exchanger reverses itself, takes the intracellular sodium, because there's so much of it, and pushes it out. And then what happens is it brings calcium in. And that's problematic because it's bringing calcium in, and now this calcium ATPase can't pump it out because you lacked ATP. And because at this point, if the mitochondria is not working and not making optimal amounts of ATP for some reason, and we're running off of ATP from glycolysis, that's not enough to be able to run all these different basic pumps. So now calcium ATPase is no longer pumping calcium outside of the cell, and it's leaving calcium inside of the cell. Which then, what can excess intracellular calcium do? Number one, here's the list of enzymes, or not all enzymes, that calcium can increase. It can stimulate phospholipase. Lipase, ASE, is an enzyme that breaks down phospholipids. Where do you find phospholipids? Oh, I don't know. Maybe around the entire phospholipid bilayer membrane. Maybe around every single organelle inside of the cell. So if you now are increasing the activity of these phospholipases, you're going to start to break down phospholipids. And if you start to break down phospholipids, guess what happens? We can start to get leaky cell membranes, or leaky organelle membranes, and I'll talk about lysosomes in a second. Well, that's a problem because I just said there's a whole bunch of stuff outside of a cell and not as much inside of a cell. And if you have an open door, then that stuff can come rushing in. Like, for example, calcium. Well, we already had enough calcium in the cell, so we're going to start to bring more in. And here's another one, is guess what can leak out? Is if you have any ATP can leak out. And if you get bored and you want to look this up, look at the function of extracellular ATP. Extracellular ATP is a signaling molecule, which would make sense potentially for the immune system, because if this cell, why would you have ATP outside of a cell? Because maybe this cell is damaged, it's dysfunctional. And if this cell is dysfunctional, then we want to call in the troops, we want to call in the immune system. So what's a good way of doing so? Maybe something that leaks out of the cell can say, hey, there's a cell that has a problem here, let's call in the immune system, and we'll take care of this cell, but might take care of cells around it. It might also be the stimulus for cell dysfunction around this, leading from cell dysfunction now to tissue dysfunction. And if this starts to go on for kind of a while, then you can get organ dysfunction, organ system dysfunction, and organism dysfunction in people that have signs and symptoms of some kind. So you can get a membrane damage. Too much intracellular calcium also stimulates the activity of proteases. These are enzymes that break down protein. What proteins? How about cell membrane proteins, and you get more leaky membrane. How about the cytoskeleton? The cytoskeleton is just a whole bunch of proteins, and now we're activating these proteases because of too much calcium, because we didn't have ATP for the calcium ATPase to take calcium out of the cell, because we had too much sodium inside of the cell, so that the sodium-calcium exchanger was bringing calcium inside of the cell. We couldn't get rid of it. Why did we have too much sodium? Because the sodium-potassium pump wasn't working, because we had ATP depletion in the first place, and because glycolysis, anaerobic glycolysis, is leading to pH shifts and not enough ATP to actually run these things, okay? Proteins are being broken down. Endonucleases. These are enzymes that are going to start to damage or act upon the nucleus. Well, we'll see what happens with that in just a second. Also in the mitochondria. Now, I'm going to slow down for just a second. Excess intracellular calcium causes mitochondrial dysfunction. I'll say that one more time. Excess intracellular calcium causes calcium dysfunction, causes the calcium to actually open up, if you will, these things called mitochondrial permeability transition pores. Again, mitochondrial permeability transition pores. Found on the inner mitochondrial membrane. The inner mitochondrial membrane, and I teach this in the mitochondria course, is really tight. These cardiolipin phospholipids that have 4 tails instead of 2. Really, really tight. And I know this is a lot, but there's a point to all this domino effect. When you start to open up the inner mitochondrial membrane, all sorts of stuff can leak out. Things like reactive oxygen species or hydrogen peroxide, which these free radicals, reactive oxygen species, can cause damage to lipids, can cause damage to proteins or glycoproteins. Hydrogen peroxide is a signaling molecule for potentially apoptosis, for this whole cell to just collapse on itself. All right, so too much, and I'll write this now, too much intracellular calcium activates or stimulates more phospholipase activity, more protease activity, more endonuclease activity, as well as tells the mitochondria to open up their channels or pores and start leaking some of their contents out, which you can tell is going to cause more damage as well. Another thing that ATP depletion does, now we saw endonucleases are going to mess up the nucleus potentially a little bit, but if you remember, the endoplasmic reticulum, there's two. There's a smooth by itself, and there's the rough endoplasmic reticulum that's associated with the nucleus. Now, on the rough, or what made it rough, were ribosomes. So now we have these ribosomes on the rough endoplasmic reticulum that start to detach. If you have ATP depletion, or the nucleus starts to become dysfunctional, then the ribosomes are going to start to detach, and then we start to get issues with protein synthesis. And this is all because of what? Oh yeah, ATP depletion. So now we start the ribosomes to start to detach, so now we get protein synthesis issues, because if you remember, the nucleus has the ability, it has recipes for every single protein that this cell might ever need to make. It doesn't make it. It takes a photocopy of it, sends it out to the ribosomes, who then translate it and take the amino acids and put them in the right sequence to make the protein in an overly simplified way. But now that we get the ribosomes popping off the rough endoplasmic reticulum, you start to get protein synthesis issues. Well, what proteins? Proteins are enzymes. Proteins are antioxidant enzymes. Proteins are channels and pumps and all sorts of things that this cell needs, cytoskeleton, in order for this cell to be able to maintain itself. Also, if you have these ribosomes that were a part of the endoplasmic reticulum that are now not a part of the endoplasmic reticulum, there's something called protein misfolding. Now, proteins are just a sequence of amino acids, and these amino acids have different characteristics. Some are more acidic, some are more basic, some are more hydrophilic, some are more hydrophobic, and because of these different characteristics of amino acids, that when the proteins are made, it tends to fold into a three-dimensional shape. And so if an amino acid is made inappropriately, or if the pH of the cell is not proper or optimal, then you might get what's called protein misfolding. And all that really means is the protein is not made correctly. Well, if a protein is not made correctly, or enough of them, then that's going to stimulate something called the ubiquitin proteasome system or pathway. And this ubiquitin proteasome pathway is basically the protein waste removal system. It's great when you have dysfunctional proteins, which is what's going to stimulate this. But there's also other proteins that, if this system is activated, is going to break them down too. And as you can see, we're breaking down proteins in the membrane and the membranes of the different organelles, for example. But for the lysosomes, if you remember, lysosome was also sort of involved in waste removal. Lysosomes have a low pH on the inside. It's said that they have dozens and dozens of enzymes on the inside of lysosomes that are designed to break things down. Just as a side note, CoQ10 is said to help maintain the proper pH inside lysosomes, which is just cool. Lysosomes, it's also said they have something like 50 membrane proteins. So if the ubiquitin proteasome system is activated, if we have reactive oxygen species that are spilling out, if we have things that can damage these proteins, well, guess what? Now, the internal contents, these 50 some odd enzymes or 60 enzymes of these lysosomes can start to spill out and start negatively impacting all the inner contents of the cell. If the ubiquitin proteasome system starts to attack the proteins that are in the mitochondrial membrane, we get more spillage of things like reactive oxygen species, maybe ATP. Who knows what's spilling out at this point? You can see that at some point this is irreversible. Well, and in fact, if you start to damage the proteins in this way, it's probably irreversible at this point. And so you can see that all these processes taking place is cell dysfunction. In the previous video, we talked about what causes cell damage or cell injury or cell dysfunction, and this is the how. How does this happen on the inside? And I'm going to pop up that slide one more time for you in just a second. But if this continues on, and already it's a bad situation, then you get necrosis or cell death. This cell, if you go back to the video I talked about, the previous video, that if what caused cell dysfunction is removed, and the cell has the proper nutrients and all the substrate and all the necessary items in order for it to repair itself, it can repair itself back to a healthy cell. But at some point, it's irreversible, and irreversible cell injury leads to cell death or necrosis. And a cell doesn't just die by itself. Typically, it's going to take some cells down along with it because of the contents, all the stuff that's spilling out of this that might impact some of the surrounding cells. Now, let's just do a real quick review, and then I'm going to show you that slide again to show you it doesn't matter where this process starts. That once it's started, that all these other things get impacted. So once again, I started with ATP depletion. If you don't have enough ATP, sodium, potassium, ATPases will be affected. That's going to lead to an accumulation of sodium inside of a cell. Water is going to tend to be drawn in. You're going to get cell swelling, and again, according to that paper, small alterations or fluctuations in cell hydration status leads to fairly significant changes in biochemical pathways as well as genetic expression. Also, if you have this happening, if you're lacking ATP, then you're going to start to decrease ATP, but it's still being used by things like they're trying to be the sodium potassium pump, and we're going to end up with higher amounts of AMP. Then that's going to stimulate anaerobic glycolysis, which is converting glucose not into pyruvate because you didn't have enough ATP, so ATP couldn't, you didn't have enough ATP, so now pyruvate probably doesn't have anywhere to go, so instead, in substrate-driven pathway, lactate dehydrogenase, pyruvate's going to shift down into lactate. That's going to lower the pH, increase the acidity, and change enzyme activity inside of a cell. So now we're running off of this very poor 2 ATP per molecule of glucose pathway called anaerobic glycolysis, which is also shifting into lactate, causing pH shifts inside the cell. Now we have too much sodium inside of a cell, and so this sodium-calcium exchanger, which normally takes sodium from outside of the cell, puts it inside, takes calcium from the inside and puts it on the outside, both of which are good because when the sodium comes in, it's going to get pumped out, the sodium-potassium pump. Now that can reverse itself, and you have so much sodium on the inside because the sodium-potassium pump's not working well that you're going to tend to take sodium and throw it out of the cell but bring calcium in. The problem is if you lacked ATP, which is what started this whole problem in the first place, then you're not going to be able to pump that extra calcium out of the cell, and it's going to stay in the cell. So via this pathway and this pathway, we're going to start to lead to too much intracellular calcium, which is going to stimulate phospholipases to break down phospholipids of the phospholipid bilayer membrane, as well as phospholipids surrounding many of the different organelles, and allowing them to potentially leak out some of their stuff or cause leakage in the cell membrane potentially as well, enough cell damage. You might get more calcium that comes rushing in Anything on the outside that has a gradient, maybe ATP is going to go out and stimulate the immune system to come to the scene. You're also going to stimulate proteases, breaking down cytoskeleton, breaking down the proteins found in certain phospholipid membranes, endonucleases for the nucleus, as well as opening up the mitochondrial permeability transition pores, which is going to allow certain things from the mitochondria to sneak out into the cytosol. Things like reactive oxygen species, hydrogen peroxide is going to further cause more damage to the cell. Then, also with ATP, and possibly the endonucleases, possibly nuclear damage at this point, can cause protein synthesis issues. Now we're not able to make as many proteins to repair, by the way, some of these things that were being damaged in the first place. So now we're getting irreversible damage because the proteins are being damaged and we're not able to make them anymore. And because of that and other reasons, the ribosomes detach themselves from the rough endoplasmic reticulum. Now we get protein misfolding. It's going to stimulate the ubiquitin proteasome pathway, which is going to start to break down even more proteins, the waste removal protein system, and we might get some spillage of things like the lysosomes, all those different enzymes out into the cytosol, which now has a lower pH because of anaerobic glycolysis, maybe stimulate more release of different contents inside of the mitochondria. And if this goes on long enough, it's going to be irreversible and you get cell death. Now, going back to the levels of organization, cell dysfunction leads to tissue dysfunction, organ system dysfunction, and organism dysfunction. And at some point, that person's not going to feel well. When enough cells are damaged, it's going to cause symptoms. So let me show you that slide real quick to see if any of these things take place, the rest of them are going to take place. So start, for example, with ATP depletion. What can cause that? You can get ATP depletion from, and what I'll do is I'll show you the cell blueprint. Is ATP depletion can be caused by mitochondrial dysfunction. It can be caused by micronutrient deficiency, that you can't make ATP well. It can be caused by infection. It can be caused by xenobiotic exposure. It can be caused by hypoxia or oxygen deprivation. You can get lack of ATP. Well, here's a good one. I'll show this to you in the next video. Is that too much glucose, according to one paper, suggests that you can create relative hypoxia. You get too much glucose coming in, and that uses up all the oxygen, that actually causes more reactive oxygen species and blocks oxygen from coming in. So too much glucose, they postulate, causes a relative hypoxic situation inside of the cell because of over oxygen consumption and blocking its ability to come in, which is pretty cool. Anyhow, glucose dysregulation, mitochondrial, can cause mitochondrial dysfunction, micronutrient deficiency, oxygen deprivation or hypoxia or lack of blood flow, for example, or anything that causes a lack of anemia. Xenobiotic exposure, infections can cause this as well. pH shifts can probably cause it, too much intracellular calcium. So you can see that just causes ATP depletion. What can cause mitochondrial damage? All the same stuff. Viral infections and bacterial infections. I showed a bunch of papers in level 3 on this. Hijack the mitochondria. That's how they do their damage in the body very often. Xenobiotics cause direct mitochondrial dysfunction, for example. Some of the other things are things like calcium entry. What can cause calcium entry to cause mitochondrial dysfunction? Anything that disturbs the phospholipid membrane, for example. So you can see that list that I showed you, misfolded proteins or membrane damage or too much reactive oxygen species, again, micronutrient deficiencies can cause that. It doesn't matter where it starts, they all lead to each other. If you start with too much reactive oxygen species, that can cause things like cell damage, or that can cause damage to the macronutrients, lipids and proteins and glycoproteins and glycolipids inside of the cell. So you can see that it doesn't matter where that starts, it's a domino effect, and all of these things lead to the other ones. And just to end this video, this is why the cell blueprint was created. Because as a practitioner myself, I was very frustrated. A patient would come in, they had a whole bunch of symptoms, and I didn't know where to start. And I didn't know where to start because it depended on who I listened to at the time, what guru or what expert. Start with the brain. Everybody has brain dysfunction, and therefore you have to start with the brain. Or everybody's toxic, so you need to detoxify everybody. Or everybody has gut dysfunction, you need to fix that. Or they have SIBO, or they have micronutrient deficiency because they're not eating well and the soils are deficient. dysfunction. Or you need to start with their thyroid, because everybody has hypothyroid. Or maybe it's food sensitivities. Everybody has gluten. Everybody's sensitive to gluten. Or heavy metal toxicity. Or it doesn't matter. Adrenal fatigue. It depends on who you're listening to. That's typically where you start. But the cell blueprint was created because it's designed as a navigation tool to, if this is all going on in somebody, the question is, where do you start? The question is, what is the thing that is the major driver for them? Is it dehydration in somebody? Or is it glucose dysregulation? Or is it micronutrient deficiency? And the purpose of the cell blueprint is to take all these things that are probably happening simultaneously and to narrow it down into the thing that's most likely driving that case, where if you address and focus on that, then many of the other things will tend to go away. So in the last video, the next video, I'll show you some specifics about how things like xenobiotics or excess free radicals or too much or too little glucose or fatty acids can drive this. But once you know this, you can do that yourself. You can look at a paper and say that viruses hijack the mitochondria, or bacteria do, or that micronutrient deficiency or hypoglycemia. It doesn't matter. That once you understand how all these things connect and interconnect with one another, you can see that it doesn't matter where it starts. Once one of these things gets going, then typically all those things will get going, leading to cell dysfunction, cell death, tissue dysfunction, and ultimately the levels of organization. The cell blueprint, why people are sick in the first place, is because of cellular dysfunction. And instead of being hypothyroid, gut, neuro, axis, adrenal, infectious disease experts, we just have to be cellular experts. So anyway, I hope you enjoyed this video, and I will see you in the next and last video in the cell blueprint.